Good afternoon, everyone. Thank you for attending today's seminar, and I want to welcome all those who are listening online. My name is Nancy Holm. I'm Assistant Director here at ISTC and the seminar organizer along with Beth Muszewski. Our final seminar for this semester will be in 2 weeks on April 27th. We'll have a speaker here from Grand Valley State University, Dr. Alan Steinman, and he'll be discussing his work on lake and wetland restoration and nutrients and contaminant issues. So please plan to join us for that talk. And in case you can't attend in person, all of the seminars are shown live via GoToWebinar and also are recorded and archived on our website. You can find the registration information for viewing the webinars on our ISTC website calendar of events. So to begin today's seminar, I'd like to ask everyone in attendance here to please silence your cell phones if you haven't done so already. And then also we'll have you, as we usually do, hold your questions for the speaker until the end of the presentation. I'll come around with the microphone so we can record those. And then for those viewing online, you may type in your questions at any time, and then we'll answer those at the end of the seminar also. So we're very pleased today to welcome as our speaker Dr. Yujie Men, who is an assistant professor in the Department of Civil and Environmental Engineering here at the University of Illinois. Dr. Men holds a BS in Environmental Engineering from Tsinghua University, and she also received her MS degree from there in Environmental Science and Engineering. She received her PhD from the University of California at Berkeley in Civil and Environmental Engineering. Before joining the Department of Civil and Environmental Engineering here as an assistant professor in March of 2016, she served as a postdoctoral researcher in the Engineering Research Center for Reinventing the Nation's Urban Water Infrastructure at the University of California, Berkeley from 2012 to 2013, and as a postdoctoral scientist at the Swiss Federal Institute of Aquatic Science and Technology from 2014 through 2016. Her research interests are on applications of microbial communities connecting the nitrogen and carbon cycles, and on the fate of antimicrobial chemicals and the development of antibiotic resistance in the environment. She's a member of the International Society for Microbial Ecology, the American Society for Microbiology, American Chemical Society, and the Association of Environmental Engineering and Science Professors. So please join me in welcoming Dr. Men today as our speaker.
Thank you very much. Thank you, Nancy, for the introduction. It's my great honor to be here and share my research with you about biotransformation of micropollutants during wastewater treatment processes. So the focus will be nitrifying activated sludge in wastewater treatment plant and the roles played by nitrifiers during wastewater treatment plant. Before my talk, I would like to give my acknowledgement to my colleagues, previous colleagues at EOWAG in Switzerland. Partially of my work is done there. I mean, 80% of the work I'm going to present is done there. And our great collaborators in University of Vienna working on AOA and AOB, those nitrifiers, they provide pill cultures to us. And also my students working on this project here at U of I. Okay, as you may all know that micropollutants also mostly include pesticides, pharmaceuticals, and personal care products. We consume it and then they are discharged into our wastewater treatment plant. Since our wastewater treatment plant are not originally designed to remove those micropollutants, our questions remain that, okay, how persistent they are. Can they be removed or not? How persistent? Can they be biodegraded or not during wastewater treatment plant, especially during biological treatment process? If they can be biodegraded, whether this biodegradation is complete all the way down to carbon dioxide in the atmosphere, or it remains some incomplete residues in the treated wastewater and get discharged together with the treated wastewater into our environments. Okay, if that's incomplete transformation, once they get discharged into natural environments, what their ecotoxicities is to the aquatic ecosystems of the parent compounds and also of the incomplete transformation products. So those are our questions. Nowadays, most of the treatment plants have been upgraded to nitrifying active sludge. So by using nitrifying active sludge during biological treatment process, we have a higher removal of COD and ammonia because we regulate our nitrogen discharge as well. Together, people found in different wastewater treatment plants using conventional active sludge and nitrifying active sludge, they see a higher removal of those micropollutants. These micropollutants in this literature, they see a higher removal. They see this correlation. The higher the ammonia removal is, the higher the removal of the micropollutants in the wastewater treatment plants. So in Switzerland, in my group, postdoc group, they have taken samples from 10 different wastewater treatment plants, and then they spiked in 10 different micropollutants. They're trying to correlate the micropollutant biotransformation rate together with the wastewater treatment plant descriptors such as temperature, COD removal, BOD removal, and ammonium removal. And then they observed a positive correlation between the removal, this is a p-value, with the ammonium removal and the ammonium concentration in the effluent. So this also shows a positive correlation. So we have this hypothesis here that, okay, does nitrifiers really have a causal relationship with the micropollutant biotransformation? So the hypothesis is, okay, it seems like there's a causal relationship. But we know correlation doesn't really mean causal relationship. We need to demonstrate this, how nitrifiers in wastewater treatment plants play a role on micropollutant biotransformation. So these two groups are the common perception of what's in nitrifying active sludge, nitrifiers and the other heterotrophic bacteria in active sludge. Which one play a major role, or what their roles are? Okay, to test this hypothesis, to test the causal relationship between nitrifiers and micropollutant biotransformation, we have mainly two approaches. Okay, we can use pure nitrifiers, like pure ammonia oxidizing bacteria, and when we add those micropollutants we are interested in, and to see whether those bacteria, pure cultures, can biotransform the compounds. Okay, several studies have been done on AOBs, ammonium oxidizing bacteria, but our preliminary study also showed a correlation between AOA, AMO gene with the micropollutant biotransformation. However, nobody has done this pure culture AOA before. So that's a question mark whether AOA, the archaea part, can also do that. And then another approach will be inhibition study, because we can use specific inhibitors to inhibit ammonium oxidizers in the nitrifying active sludge. So if we see an corresponding inhibited biotransformation activity, we can indirectly demonstrate, okay, there's a relationship between ammonia oxidizers and the biotransformation activity. In the end of my talk, I'll talk about some removal monitoring in a full-scale wastewater treatment plant here locally, because we have a specific treatment step, which is an additional nitrification step, nitrification tower. So if we observe a removal during this nitrification tower process by actual monitoring of this full-scale treatment plant, we can also show some evidence about the roles played by nitrifiers during wastewater treatment plant processes. Okay, so why we care about AOA? Since most wastewater treatment plant in the nitrifying active sludge is AOB. AOB, the bacteria counterpart, is the most dominant. But in some cases, like coastal area, when we use seawater a lot, and also in tropic area, the AOA, the archaea counterpart, can also be dominant. But people didn't really pay too much attention to the AOAs. And also, AOA is universally distributed. They are ubiquitous, found in environments, not only in wastewater treatment plants, but also in natural environments such as farmland, soil, sediments, and seawater. Sometimes they are in high abundance in those natural environments. They have distinct physiological characteristics because AOA has a high affinity of ammonia. They can use very low concentration, even 100 to 1,000 times lower concentration of ammonia by AOA. Since we know that micropollutants, they are low in concentrations. So this might give AOA some advantage to take up those low concentration compounds and use co-metabolism to biotransform those compounds in nature and also in wastewater treatment plants. So, and also, of course, nobody has done this research before, so we didn't know whether this archaea, the AOA, can do the micropollutant biotransformation. So there's a significance. We want to demonstrate their ecological significance in the micropollutant biotransformation. Okay, so in collaboration with the University of Vienna people, we got 1 AOA strains, because currently there are limited numbers of AOA pure strains available. So this is the one we can get from our collaborator. And also to compare, we also include another AOB strain, another 2 actually AOB strains, which were isolated from wastewater treatment plant as a comparison. Okay, and the tested micropollutants include these 10 different compounds. They belong to 3 different categories: phenolures, tertiary amines, and tertiary amines. These are the structures of those compounds. Okay, out of the 10 compounds, actually, we did AOA and AOB biotransformation study. 2 of them showed biotransformation activity for both AOA and AOB strains we used. Whereas AOA strains on the top here show the higher here, the purple line. The purple curve is the AOA biotransformation curve. It shows a faster biotransformation than its AOB counterpart, the 2 AOB strains here. For meandering, it's for AOB slower, which is consistent with our hypothesis. Like, since AOA, ammonia monooxygenases have higher affinity to the substrates, so this meandering, this pharmaceutical meandering may have also bind to AMO, is a co-substrate for AMO enzyme, and can be co-metabolic transformed. The same thing for ranitidine. We can see the purple line for AOA got faster. This is the pseudo first order rate, reaction rate constant. We see this is archaea. It has a higher rate constant as well. Okay, and then we also look at the transformation products, because our second question is whether they can completely be removed or not, whether they can serve as energy source or not. So we look at the transformation products. It seems like an incomplete transformation. For meandering, the transformation product is this alpha oxo adding an oxo group at this ring. For meandering, this is structure we can identify so far, and it increases and then decreases. Seems like they're also it gets further biotransformed, but we were not able to transform the further transformation products. For ranitidine, we have found 3 transformation products which all undergo oxidative reactions. However, their concentration is relatively low. They didn't close up the mass balance, indicating that there are other unknown transformation products for ranitidine for this compound. Okay, a summary of this pure culture study is 2 out of the 10 compounds we tested have the biotransformation activity for AOA and AOB pure cultures. And also meandering undergoes this oxidative reaction forming alpha oxo meandering. Seems like very likely AMO is the enzyme, because AMO is known to carry out the monooxygenation reaction. And both biotransformation of meandering and ranitidine seems like co-metabolic, because none of those compounds can support the growth of AOA or AOB. Okay. So next, pure culture study, we show that partially the tested compound can be partially biotransformed. But there are also disadvantages of these pure culture studies, because the pure cultures we have limited access to those pure cultures. They are not so representative to the actual ammonia oxidizers in wastewater treatment plants. They might not be representative. So in order to be more environmentally relevant, we turn out to use inhibition study. We use actual wastewater treatment plant, actual sludge from the wastewater treatment plant. We add inhibitors to inhibit the AOB there in situ, locally, and then we can monitor, we investigate the biotransformation activity change. So here is the hypothesis. We use specific inhibitors to inhibit the the native ammonia oxidizers. So the ammonium oxidation is stopped, is ceased. If we could observe the biotransformation of those micropollutants, the inhibition of the transformation activity, we can show indirectly, okay, the relationship with these ammonium oxidizers and the biotransformation process. If the biotransformation process is largely carried out by heterotrophs there, we won't see any change in terms of micropollutant biotransformation. So it will be similar with the control group. So we have two different groups, one control without adding inhibitor. Inhibitor treated group is we add some AOB inhibitors. So we also use this ammonium uptake rate control reactor to control the ammonium oxidation activity to make sure in the inhibitor treated reactors, the ammonium oxidation activity is completely inhibited. Inhibitors we used 3 different inhibitors. The first 2 inhibitors are AOB inhibitors. The first one is allylurea. It is a commonly used inhibitor in engineering, in wastewater treatment plant, or in nitrification investigation. The mechanism for ATU to inhibit ammonium oxidizers is they chelate copper. Copper is an essential metal for ammonium monooxygenase. So once copper is chelated, the AMO activity is inhibited. Octine is reported as a more specific mechanistic inhibitor for AMO. It binds covalently to AMO and then inhibit AMO activity. So it's thought to be more specific, and also it's more specific to AOB species. The third inhibitor we use is PTIO. This compound, by adding this concentration, showing 100% inhibition on AOA, whereas no inhibition on AOB. Using these different specific inhibitors, we can disentangle the roles played by different ammonium oxidizing groups like AOB and AOB. We can differentiate those, AOA from AOB. So first, we took a look at the ammonium oxidation activity. This is all we want. We want to make sure the ammonium oxidation activity is inhibited. So as you can see in this graph, by adding octane or ATU, the blue and red, ammonium oxidation is completely inhibited, whereas adding PTIO, and it shows similar ammonium oxidation activity with the control group, which indicates that in this local wastewater treatment plant, in the nitrifying active sludge community, the AOA, the archaea group, is less abundant. So there's little archaea there. And this can also be reflected by this biotransformation plot. So this is the biotransformation. This is the rate constant. Y-axis is the rate constant. You can see PTL only inhibited to a small extent for meandrin and ranitidine, but not the other 8 compounds. Whereas these 2 compounds we know can be biotransferred by AOA pure cultures. This is a consistent result. But since the presence of AOA is less abundant, the biotransformation activity also dependent on the biomass, the number of the AOA culture in the sludge. So we only see a small portion of the biotransformation has been inhibited. Whereas when we look at the AOB, when we use octane or ATU as an inhibitor, we see 8 out of the 10 compounds showing inhibition. You can see here. All the final urea and some of the amides and amine group showing the inhibition. Besides the inhibition of micropollutants by AOB inhibitors, we also see there's difference between the 2 AOB inhibitors, between the ATU and octane. So if we assume both inhibitors contribute the same to inhibit AOB, we shouldn't see any difference between the 2 inhibitors. Whereas we see a higher inhibition by ATU here, the red color, than octane. Why? According to their mechanisms, we think, okay, ATU inhibit more. Since ATU is a copper chelator, is it very specific to AOB? We don't know. It's a question mark. So is our common perception correct? Because earlier studies many studies used ATU as a specific AOB inhibitors and demonstrating, okay, AOB has involved a lot in micropollutant transformation. Is that the case? Is that true? So we want to extend our compound list and to show whether this is true or not, whether AOB is really playing an important role reflected by the ATU inhibition studies. Okay, so we extended our compound list from 10 to 79, and then we used ATU and octine as inhibitors, and we compare the micropollutant biotransformation with inhibitor-treated group and the control group. Out of the 79 compounds, we see 55 got removed. More than half can be removed by nitrifying active sludge, more than 20%. This is not bad. We see they can be removed. And then this is the results. This is all the, among these 55 compounds, this is the 33 compounds that showed inhibition by ATU and octine treatment. All these compounds, all these bolded compounds showed inhibition by adding ATU or octine. Means the biotransformation of these compounds is attributed to ammonium oxidizers, whereas the unbolded compounds, they didn't show any effect by adding AOB inhibitors. So among these 31 micropollutants, we compare the inhibition extent between ATU and octine. We do this plot, we do this histogram plot, the distribution. We look at the ratio of the removal in ATU or octane added group. If the ratio is less than 0, this is log2 ratio. If it's less than 0, that means ATU showed more inhibition than octane. We see a shift. We see several compounds show the shift to the left-hand side, which means the ATU showed higher inhibition for those compounds. This is our earlier perception of the composition in nitrifying active sludge community, which consists AOB and heterotrophs. And then when we use ATU, we think ATU inhibit only AOB. And then we think, okay, AOB played an important role in micropollutant biotransformation. Now, when we do ATU experiment and octane, a more specific AOB inhibitors, we see the difference. Now, our hypothesis and our view of this nitrifying community composition changed. We updated it like this. So ATU can not only inhibit AOB, but also they can affect some other microorganisms. And these microorganisms are likely also involved in biotransformation process, particularly in the 31 micropollutants. So we are going to look at the details. The hypothesis now here become, okay, if we see a similar inhibition between octane and ATU treated group, between the two inhibitors, we are more confident to say, okay, AOB are involved. Whereas if we see compounds that's showing higher inhibition by ATU than by octane, okay, that's a question mark. Maybe part of the biotransformation activity was attributed to other ATU sensitive microorganisms rather than AOB. Of course, if there's no inhibition effect, we know that's ATU insensitive heterotrophs that carried out the biotransformation activity. We want to quantify this. So we quantify the inhibition extent using this formula, the difference between the removal of the treated group and the control group, normalized by the removal in the control group. This is the extent of inhibition. We calculate this extent of inhibition for all the 31 compounds, and we compare this inhibition extent between ATU and octane. So this part will be, this is the ATU transformation curve, this is the control transformation curve. This will be the proportion of this part will be the inhibition extent of ATU, and this portion will be the inhibition extent of octane. In this way, we we can show, using this inhibition extent, show how much the inhibited microbial members contribute to the biotransformation activity. For example, this part was unaffected. It can be attributed to the unaffected microorganisms. And this part for octine can be attributed to AOB only. And the difference between ATU and octine can be attributed to the ATU-sensitive heterotrophs. Okay, now we take a look at the comparison. We found 11 compounds showed similar inhibition between ATU and octine for the first 46 hours. So during the first 46 hours, both ATU and octine showing complete ammonium oxidation inhibition. So we didn't see, this is a nitrate formation. We didn't see a nitrate formation in both ATU and octine treated samples. Whereas after 46 hours, in this octine group, blue curve, we see an increase of nitrate, means there's a recovery of nitrification activity. Whereas for the ATU, the red color, the red curve, there's no nitrate formation. So for ATU, remember, all over the incubation period, ATU has a complete inhibition, whereas octine before 46 hours, there's a complete inhibition on ammonium oxidation activity. And after 46 hours, this ammonium oxidation activity recovered. By comparing this temporal difference, we can also demonstrate which compound's biotransformation is related to AOB. Okay. So we compare the biotransformation, the removal, the inhibition of the 11 compounds after 46 hours. You can see this blue bar here. They all showed a decrease in inhibition, means the biotransformation activity recovered together with the recovery, with the restoration of ammonium oxidation activity. This can demonstrate this 11 compounds biotransformation was attributed to AOB. Okay, this is direct evidence. This is more confident evidence. Particularly for the 4 compounds, monurone, azulone, trimethoprim, and clomazone, we can see this inhibition extent is higher than half, which means more than half of the removal of those micropollutants were attributed to AOB. Okay. So this is the degradation curve. We can see at the beginning, before 46 hours, there's almost a complete inhibition on the biotransformation. After 46 hours, as the ammonium oxidation activity restored, we can see a recovery of the biotransformation activity as shown in the blue curves in octine added samples. Whereas in the ATU added samples, there's a constant inhibition on micropollutant biotransformation. Okay. For 17 micropollutants, we see a higher inhibition by ATU than by octine. By using this inhibition extent, we can allocate how much of the removal was attributed to different microbial groups. For example, this blue color is the inhibition extent of AOB. The higher it is, the more contribution it is. The same for the red color. The left panel, we can still see, although AOB and the ATU sensitive microorganisms play some role, but more than half of the removal were attributed to the heterotrophs. Whereas on the left ones, on these ones, if we use ATU, we think, okay, more than half of the removal, even more than 80% of the removal were attributed to AOB. But actually, when we look into details, more than half of the 80% of the inhibition actually was contributed to this ATU-sensitive microorganisms. So earlier, we might overestimate the role of AOB in the biotransformation of these specific micropollutants. If we look at the compound specificity for this AOB-sensitive microorganisms, we can see this green boxed compounds, they belong to phenylurea pesticides. You can see here, this ATU-sensitive microorganisms tends to biotransform those compounds, as well as the phthalo ethers. We can see most of the phthalo ethers we targeted get transformed by this ATU-sensitive microorganisms. Okay. Also here, just I want to quickly mention this. We didn't see any inhibition effect for these compounds. So for those compounds, esters, for example, they undergo hydrolysis reactions. For the compounds chloroacetonylize, they also undergo the substitution reactions, and also sulfonamides. We didn't see a really observed inhibition for those compounds, indicating that those compounds biotransformation were not carried out by ALB or ATU-sensitive microorganisms. One exception for sulfonamide antibiotics is this sulfothioxazole. It has been inhibited by 15% by ATU. Why? Because it has this structure. If you look at the compound specificity, phthalo ethers, this structure, phthalo ether-like structure here, may contribute to this part of 15% inhibition by ATU. Those ATU-sensitive microorganisms may attack to this part and biotransform on this part to some extent. Okay, so we know this. We now know ATU might not be so specific. We might overestimate the roles played by ALB actually during wastewater treatment plants. So this table summarized the micropollutants that have been reported to be correlated with ALB using ATU inhibition studies. The ones I highlighted is reported in the literature using ATU inhibition studies. And also, we also include these compounds in our study. We show a much higher, you can see this, we can show a higher inhibition by ATU than by octine, another ALB inhibitor, which means the biotransformation of these compounds were not only attributed to ALB, but also other ATU-sensitive microorganisms. Earlier, we allocate this compound. We allocate this compound under ALB. They might be carried out by ALB, but now perhaps we need to transit it from this column to the ATU-sensitive microorganisms. However, we don't know which microorganisms they are, but we know we might have overestimate the roles played by ALB for these compounds. Okay. And in the end here, since after I moved here, I know the local wastewater treatment plants implemented a special treatment step, which is nitrification tower. It's because the weather here is cold, they add this additional treatment process in order to refine the nitrification activity in winter. So this nitrification tower is perfect for this study to investigate the roles played by nitrifiers, because in this tower, I would assume nitrifiers were specifically enriched because they take the treated wastewater where there's low COD or BOD, there are low organics. So nitrifiers can be particularly enriched in this nitrification tower. So we take samples from different treatment steps, and then we investigate the removal, the micropollutant removal, intrinsic micropollutants in the raw wastewater, the removal in this nitrification tower, whether they can be removed or not. So this is the results. We compare the different removal steps, different color. We took samples from the two wastewater treatment plants locally, one from northeast in Urbana, one from southwest in Champaign area. So each color represents each treatment step. Primary clarifier, secondary treatment process, secondary clarifier, the yellow color is nitrification tower. If you look at specifically the yellow color, you can see in this northeast wastewater treatment plant, these are the x-axis, the different categories of the micropollutants. You can see they actually didn't play very important role in the biotransformation, in the removal. Although we know in the biological treatment process, there's also nitrification activity there, but there's also some heterotrophic activity together combined. So you can see the majority, the removal, were contributed largely by secondary biological treatment process. similar with the other treatment plants. We can see a higher removal for this metabolized and industrial chemicals, but also very relatively low removal contributed by nitrification power. Okay, another reason might be after secondary treatment, the micropollutants concentration is rather low. It could be another reason to explain this low removal observed in the nitrification tower. But in the end, my point is, for pure culture, we know, okay, of course, AOA and AOB, pure cultures can biotransform, but only part of the micropollutants we tested for meanser and ranitidine via co-metabolism by ammonium monooxygenase. And ATU is actually not a very specific AOB inhibitors. We might have overestimated the roles played by AOBs if we use a more specific inhibitor like octine. Okay, the AOB in nitrifying active sludge do play a major role, but to a smaller group of compounds like azolone, clomazone, monorone, and trimethoprim. But these still need to be further tested by pure culture studies and by more sophisticated design of study to be further demonstrated. And then other microbes with ATU affected or ATU sensitive enzymes might play important roles for some compounds, for example, thiol ethers or phenylurea groups. And AOB and microorganisms affected by ATU were not involved in these compounds like esters, chloroacetanilides, sulfonamide antibiotics. In the end, I want to mention is that from this study we see, okay, AOB do play some roles, but the removal of micropollutants during wastewater treatment plants is a combined effect by both heterotrophic bacteria and autotrophic AOB nitrifiers. So it's carried out by multiple community members, maybe undergo different transformation pathways. So to further disentangle the transformation products, identification will help to differentiate the roles played by different community members in order to help us to more accurately estimate the fate of the micropollutants once get released into the natural and engineered environments, where including nitrifiers or other heterotrophic microorganisms. So this is the take-home messages for this study, and I would be happy to take your questions. Thank you very much.
Yes. Okay, thank you very much. We'll take questions from here at our audience right now if we have some.
Thank you for your talk. In slide number 29, where you compared the two wastewater treatment plants, one of the primary unit processes that come up in the Southwest treatment plant is the anaerobic digester, the reactor. So you mentioned that the heterotrophs and the autotrophs are playing a role. And how do you compare, the role of the digester and what communities are, contributing to the removal?
Oh, that's anaerobic. That's an EPPR. That's an enhanced phosphorus removal reactor.
Mm-hmm.
So we haven't identified the microorganism there, but that's a good question. We have studies showing that micropollutants can be transformed under both anaerobic and aerobic conditions. They must have undergone different pathways. So that's a good question. They can be both. They can be removed under both conditions, both redox conditions, but they must undergo different removal processes, removal pathways. And we were looking at that now. Yeah. Thanks.
Okay, are there other questions here from the audience?
I think I was looking at slide 23. No, it was the one with the relative contribution of the, yeah, there it is.
So in order to do this kind of analysis, it seems like you have to assume that the effect on the different communities is the same, and it's the same over time. In other words, if some microorganisms are 50% inhibited and other microorganisms are 100% inhibited, is that embedded? Do you have to make that assumption to do this analysis?
Sure. That's why we look at the 46-hour inhibition, because we know before 46 hours, both inhibitors like ATU and OCT, they inhibited completely, at least for AOB, but we didn't know the inhibition effect for other microorganisms because we cannot monitor. That's a good question.
Right. So the heterotrophs could be
we have to assume,
much more different effect. So
right. We have to assume the inhibition, especially I would say OCT would be more specific. We can say, okay, OCT can only inhibit AOB, but ATU for those microorganisms, we can only assume, okay, it's also 100% inhibition.
Okay.
For the other.
Okay. And then I'm just curious, for all of the things that you've looked at for partial breakdown products, because it looks like the breakdown is not nearly complete. It's pretty small metabolic steps. I'm just curious, are you concerned about any of the breakdown products?
You mean further breakdown? You mean, the toxicity of the transformation product?
Well, yeah, so yeah, if you only partially break them down, are there any of the breakdown products you're more worried about than the parent products?
There are cases, but I know there's that we didn't do toxicity tests because, but I know there are studies looking at the overall toxicity before and after wastewater treatment plants. They showed an overall decrease of the toxicity. That's overall. But for specific compounds, it's hard to say. It could be for some compounds, the transformation products is even more toxic than the parent compound. But that's a good question. But in general, the overall toxicity is decreasing according to previous studies, but we didn't do the toxicity test ourselves.
Okay, thanks.
Do we have any other questions here? Okay, and I don't believe we have any online. So I'd like to again thank our speaker for today for a very interesting presentation. Thank you.